In this segment, we're going to go over the viral replication cycle as described in Chapter 13. What I really hope that you'll be able to do is to understand the basic replication cycle, but then compare it to the replication cycle of viruses like HIV and SARS-CoV-2. These viruses have variations compared to the general cycle. It will be important to be able to compare and contrast these viral life cycles. First, it's important to remember that the virus is very dependent on the host cell to do all of the viral replication. Typically, the replication is going to end with the death of the cell. Depending on the virus type, the cell death may come very rapidly or take longer. We'll discuss this in detail as we move through this section. In this example, we're looking at a bacteriophage, much like the one that we work with in Microlab. The steps of viral replication are shown here in bold. First is attachment, then entry into the cell, followed by synthesis of all the viral components. Assembly of new viral particles comes next, then release. If we look at this animation that I drew a long time ago, you can see how this works. If we're looking at the first step, the virion has to attach to the host cell, and it does that with the tail fiber, which is the viral ligand, which binds to the host cell receptor. Then, in this case, the genome gets in, the entry part. The virus actually injects first enzymes to break down the cell wall, and then the enzymes are followed by the genetic material, which will pass through the inner membrane channels of a bacteria like E. coli to get into the cytoplasm of the cell. After viral entry, the virus has to make new viral particles. The cell actually helps now since the viral genome serves as the instructions to synthesize new virus components. The virus needs both protein as well as copies of the genetic material. The cell does make both for the virus. Once you have enough of the components, they assemble as new viruses. This is a spontaneous process. You can think of it like crystallization. Something similar happens if you have a bunch of sugar and water and then you drop something into the solution like a stick. As the water starts to evaporate, you'll get little crystals forming on the stick, and then all the sugar crystallizes out over time. It's a spontaneous reaction. The same thing is happening with the virus. It's spontaneous. Once the concentration of viral components is high enough within the cell, they'll assemble into new viruses. This is kind of interesting to think about. An analogy to building virions in this way might be building a gingerbread house spontaneously. Imagine that you're in your kitchen and you're building all the little pieces of a gingerbread house, the walls, roof panels, doors, window frames, and then when you have enough of it in your kitchen, the pieces just kind of spontaneously come together and form your little gingerbread house. No manpower needed. This is similar to the virion assembly in the cytoplasm. Now, the virions have to get out of the cell. Depending on the virus and cell type, the cell either is going to break open, lyse, and all the virions spill out, or the virus pushes its way out of the cell, taking lipid bilayer and protein with it. Breaking open the cell is called lysis, and pushing out, taking membrane with it, is called budding. Budding is how an enveloped virus is formed. Let's talk about this cycle with another twist for some bacteriophages. This cycle should seem a little familiar to you because we were talking about it as we discussed transduction in Chapter 7. However, it's important to point out that this is the regular lytic cycle of a T-even bacteriophage. Here the virus attaches and then enters by injecting its DNA. During the infection of the cell, the virus replicates and the bacterial chromosome degrades and breaks apart as new copies of the virus is made. Then when you have enough of all of those components inside the viral particles assemble, which then break out of the cell. This process describes the lytic type of viral life cycle. At the bottom of this slide, you can see a little animation that's depicting spontaneous assembly of viral particles. That would be happening inside a cell. If we look at this graph, you'll see how this kind of lytic infection is depicted. If you look over here on the y-axis, it reflects the number of intact virions, meaning viral particles that are capable of infecting a cell. If you start with just a single virion, when that enters into the cell, there will not be any infectious virions because now there are only genome of the virus inside the cell. If you were to break everything open and look for virus at this point, you will not have any infectious virus. You only have viral genome, incapable on its own of infecting a different cell. Then there's going to be a period of time while the virus is replicating the components it needs before it assembles new virions. Once the new virions are assembled, then they will break out of the cell. There is a mass release. All the virions emerge at one time, and then they'll start a new cycle of infection in nearby bacteria. The period of time from initial infection to release is called burst time. It's measured from the time of initial infection to the point where viral particles are detected in the medium. It's the time required to replicate virus. Remember, though, that for some types of bacteriophages, we have the ability to enter into a lysogenic cycle, which is part of the cycle where the phage integrates itself into the chromosome. The bacteriophages, bacterial viruses, capable of entering into a lysogenic cycle are called temperate phages. When they integrate into the bacterial chromosome, you refer to the phages as prophages. Prophages are the bacteriophages that are sitting inside the bacterial chromosome. They've integrated into the chromosome. An interesting fact is that sometimes those prophages bring extra genes with them from their previous host. This is a version of transduction event, where the bacteriophage brings genes from the previous host into the new host. When that happens, it's called lysogenic conversion because the lysogenic phage is carrying genes that alter the phenotype of the host bacteria. They convert them. Sometimes these extra genes are toxins to the host cell. For example, have you heard of the infection scarlet fever? What about diphtheria? Lysogenic conversion has occurred with those two infectious bacteria, and the viruses infecting the bacteria give the bacteria a new phenotype. In this case, dangerous ones, because it causes a disease in humans. Only because the bacteria is infected with the virus that brought an extra gene along, a gene that encodes a toxin that affects human health, do the bacteria themselves cause an infectious disease in humans. In the last part of the semester, we'll talk about infectious diseases. We'll come back to the lysogenic conversion topic. So this figure shows the relationship between the lytic cycle and the lysogenic cycle. Sometimes when a phage injects its DNA, the DNA incorporates itself into the chromosome rather than entering directly into a lytic cycle. That integrated phage will just replicate with the bacteria. Every time the bacteria divides, it also replicates the viral DNA as well. Then, at some point, there'll be some sort of event to trigger the virus to exit the chromosome. That event is called induction. It might be a stressor, but something happens that encourages the phage to emerge from the chromosome and enter into that lytic cycle. The lytic cycle ends with the bursting of the cell, releasing virus. This figure is illustrating that relationship between the lytic and the lysogenic cycle. Please remember that this is a normal relationship when normal infectious bacteriophages are being produced. In Chapter 7, we were looking at events that occurred during these cycles that were a mistake that happened either in the lytic cycle, which would lead to a generalized transduction event if any random piece of the degraded chromosome was packaged instead of viral genome and moved to a new host incorporated. That's generalized transduction. Remember that it's a very rare event because normally the viruses get packaged normally and moved to produce a new infection. If you consider the lysogenic cycle, and if when the virus tried to excise to emerge out of the chromosome and enter the lytic cycle, sometimes the virus might accidentally leave a bit of the virus behind and take a piece of bacterial gene near the virus when it was in the chromosome. If this hybrid form of the virus moves to a new host and is incorporated into the chromosome, That was specialized transduction. These were the mistakes that led to transduction, whereas this particular slide is just showing the normal, no mistakes made, no transduction occurring type of events. When we consider animal viruses, they have the basic replication pathway as the phages. The steps of attachment, then entry, then synthesis, and maturation, then release, all of that is generally the same. But for some of the animal viruses, they have an envelope, the lipid bilayer that is around some of the viruses. Furthermore, keep in mind that eukaryotic cells are different than prokaryotic cells. For example, animal cells do not have a cell wall. Animal viruses don't have that tail or tail fiber type of arrangement seen in bacteriophages. They do have those glycoprotein spikes. That coronavirus that we spoke of in the last video has glycoprotein spikes, and in the envelope that will allow the attachment to the host cell. The attachment is mediated by cell receptor and viral ligand. When an animal virus enters, it uses one of the three mechanisms depicted in this figure from your book. The first they call direct penetration. Basically, after the virus attaches, it uses a pore or a channel that's on the membrane of the cell, and it shoots the genome right through that pore. That is direct penetration, using a channel to get in. Another mechanism of entry is endocytosis. Pretty much any virus could use this mechanism on an animal cell. Endocytosis is basically when the cell folds the membrane internally, kind of pinches in a piece of the membrane and forms a vesicle. It's a way of internalizing substances outside of the cell. Once the virus is attached to the surface of the cell, that cell is somehow stimulated to pinch in that piece of membrane inside to endocytose the virus. Now you have this little vesicle that has been brought into the cell and the virus is inside. Then the membranes, both the cell membrane and the viral envelope, if there is one, is going to be dissolved away. The capsid too is dissolved. It's broken down by cellular enzymes of the host cell. Now the genome is inside, exposed, capable of serving as the instructions to build more virions. The third mechanism of animal virus entry is membrane fusion. That's very specific to enveloped viruses. Some of the enveloped viruses use this mechanism. Fusion is when the two membranes, the envelope membrane of the virus and the cell membrane, are brought into very close proximity so they actually fuse together. It's similar to when you gently push two soap bubbles together and they fuse into a larger bubble. That mechanism of fusing the two membranes together dumps the viral capsid inside of the cell. Again, the cell dissolves the capsid to expose the viral genome. Those are the three mechanisms of getting an animal virus inside the cell. The replication process is going to be a little different depending on the type of nucleic acid inside the virus. If it's a DNA virus, it's typically going to head to the nucleus and use the machinery of the host cell to transcribe RNA and translate to protein. If it's an RNA virus, though, it's often replicated in the cytoplasm because most of the machinery is there. However, since animal cells do not replicate RNA, the RNA viruses will definitely have to use their own polymerase. RNA viruses, they're going to have to do their own special polymerase to deal with the RNA genome because our own cells don't deal with replicating RNA. Eukaryotic cells make RNA from DNA, but they don't replicate RNA. It's an important point for viral replication to consider how messenger RNA is going to be synthesized. What's the template for the genome? This chart is not for memorization, but it is a comparison chart from your chapter and shows all of the different types of genomes and what the virus has to do in order to allow the cell to make message. Obviously, the virus has to make messenger RNA in order to make its proteins. New virions need protein, and they also need genome to package. What the virus uses as a template for a genome is also shown. If you look at the first two rows in this table, you'll see that these these DNA viruses don't do anything too special or unfamiliar because our own cells use DNA as a genome. Animal cells, such as our own cells, are capable of replicating the virus just fine. However, when we're talking about RNA viruses, our cells do not copy RNA. The virus has to encode, or come packaged with and encode, an enzyme that can do something with the RNA. These last 4 rows are describing different types of RNA viruses, whether it's a single-stranded RNA virus or the viral genome is double-stranded. The plus or the minus sign is referring to as sense. The sense refers to whether that RNA looks like messenger RNA or not, whether the RNA genome can be translated directly into protein, or if a complement of the genome is needed to make protein. The positive sense is basically messenger RNA. If a virus has positive sense RNA, then it's able to be translated immediately into protein. Ribosomes can sit down and translate and build viral proteins from the sequence. Whereas if you have a negative sense genome, like here, then you have to have an enzyme, a polymerase, that can copy RNA, come into the cell with the virus so that it can make a complement of that single-stranded piece of RNA. The complement of a negative sense is now used as messenger RNA. This figure from your book is describing that replication cycle between the different viruses. Let's break this down. If you look here in this first panel, you have a positive sense single-stranded RNA. Then that RNA genome is a messenger RNA. You can see that immediately it can be translated into protein. Then you also make a complement of it as a negative sense RNA template that then goes through another complement to make positive sense again. The negative sense is serving as the template to make more of the viral genome. Really what happens is that the viral polymerase makes complements of the RNA genome and then makes a complement of the complement. Remember, to deal with RNA viruses, the virus needs to have special enzymes encoded. Animal cells will not replicate RNA. If we examine this negative sense RNA virus, it will have the requirement that the RNA polymerase is packaged with the virus. This is needed so that you can make a complement of the negative sense into positive sense that is then used as message to make viral proteins. A positive sense can be translated directly, but the negative sense must be copied first before translation can happen. The positive sense will not only be translated so viral proteins can be produced, it also serves as a template to make more negative sense RNAs, which are the genome. You need copies of the genome of the virus before you can make functional infectious variants. Assembly, shown down here at the bottom of these panels, is the making of new virus particles. Double-stranded RNA viruses will unwind so that you have positive and negative sense. Of course, as we mentioned, the positive sense is used as message, but it also serves as the template to make the negative sense. The negative sense serves as the template to make positive sense. We're going to have to use both of these in order to make more genome of the virus. The bottom line is, when you're dealing with RNA viruses, you're going to have virus-specific enzymes, virus-specific polymerases to deal with this need to replicate RNA. Our own cells, all eukaryotic cells, do not make copies of RNA. If we have an RNA virus, then we have to have a virus-specific polymerase capable of creating complements of the RNA. A thought that ties in well at this moment is to draw a connection between these viral-specific enzymes and therapies. When you have a molecule like an enzyme that is very specific to the virus, like these polymerases that copy RNA, these enzymes are unlike any within our own cells. These unique enzymes become a good target for potential drug therapies. Drugs might potentially bind to these enzymes and interfere with viral replication, but have no effect on the host cell. This is the ideal type of drug. When it comes to assembly and release of the viruses, different scenarios exist depending on the virus. If the virus is a DNA virus and it's actually being synthesized in the nucleus, it could bud out of the nucleus into the cytosol and exit the cell. RNA viruses develop in the cytoplasm and they potentially bud out from the cell membrane. But the envelope viruses acquire the envelope, which is membrane, from their host cell. Envelope viruses tend to cause longer-term infections that are more persistent because they don't kill the cell right away. The cell lives for a while producing these viruses before the immune system catches up with it and kills it off because it's infected. Or the cell finally collapses from the exertion of making so many viruses. Over here's an animation showing how a virus pushes its way out, buds from the surface of a cell. This is an animation intended to give you a visual for how that works. Naked viruses are mostly released by exocytosis from animal cells, although they could potentially cause lysis and death similar to the bacteriophage that we were looking at. Next, I want you to compare this graph to the one that we looked at previously concerning the burst time for bacteriophage. Remember we observed that very sharp increase of virion particles in the medium because the cell lysed and released everything, all the virions at one time. And these animal viruses that are budding out, it's a slower accumulation of infectious virions in the medium. You can see an increase over a more extended period of time. They're budding out, not lysing and releasing all at once. Next, I want to discuss the life cycle of a specific animal virus, HIV, and compare it a bit to the basic viral life cycle as discussed with bacteriophages. There are many similarities, but there are also unique differences specific to this virus, as with all viruses. You have been linked to a narrated animation explaining the HIV life cycle. Do learn it. For now, we'll just look at a simplified version of the HIV life cycle. HIV is an RNA virus from the family of retroviruses, meaning that the virus starts out with a genome of an RNA, but when it gets inside the host cell, it converts it to DNA. As DNA, it then integrates into the chromosome of the host permanently. This is a retrovirus. HIV belongs to an RNA retrovirus family. When you look at this image here, you can see that like all viruses, HIV must attach. The virus uses its ligands to attach to specific types of cells. Then HIV performs a membrane fusion to dump its core, its nucleocapsid, inside the cell, which then gets disassembled. Then the genome, which is RNA, is converted to DNA, and it enters into the nucleus and incorporates into the host genome. RNA copies are made from that viral DNA, which serve as both message and as genome of the virus. Those RNA copies will, after transcription, leave the nucleus and enter into the cytoplasm, and then translation occurs to make new viral proteins. Those viral proteins congregate at the surface of the cell. Then the nucleic acid starts to assemble parts of the virus, and this pushes its way out. It buds out. The virus is interesting in that it doesn't really mature until after it gets out of the cell. It fully matures after it leaves the cell. These are the basics, but there are more details discussed in the animation. Please watch the animation on the HIV life cycle and learn the specific ligands, receptors, and the enzyme names. The life cycle of HIV is very similar to the bacteriophage life cycle. You can see there is attachment, entry, synthesis, assembly, release. However, there are some specific details that differ. Please watch the animation and pick out those specifics. In the last few minutes of this presentation, we'll go over a viral characteristic called latency. There are only a few types of viruses that can do this. The herpes family of viruses is famous for undergoing latency. Latency is similar to hibernation. The virus stays put in special cell types after infection. It doesn't get cleared out, but it doesn't do anything in the cell for periods of time. This is latency, the quiet phase. Then the virus will reactivate, multiply in cells, and kill them and cause disease. The herpes family of viruses includes the virus that causes chickenpox, which goes latent and reactivates as shingles, herpes simplex viruses, causing very painful blisters that go away only to reemerge. These blisters caused by the herpes simplex viruses are known either as cold sores if they appear on the mouth, or genital herpes if on the genitals. These viruses undergo these periods of latency and reactivation. The virus sometimes incorporate into the genome of the host as well. If they do, instead of being referred to as a prophage, they are proviruses. They are not phages after all. They are animal viruses that are incorporated into the DNA, and that's permanent. They don't come back out once they go in as a lysogenic phage might. These viruses are not undergoing lysogeny, where they could potentially come back out and cause some activated infection. Once they go in, they stay there. We'll be talking more about the herpes family of viruses later in the semester. But since we're talking about viruses in this chapter, it is important to introduce this concept of latency and proviruses. Now, it might be a good place to stop this presentation. We'll pick up with viruses in cancer in the next presentation.